Welcome back to our flatworm series. Hi, I'm Christopher. In the last video, we gave Echinococcus the serious One Health spotlight it deserves. Now we move on to large animal and bird cestodes. The organizing rule is simple. Start by asking what kind of host you are looking at, and just as importantly, what role that host is playing. Horse, ruminant, bird, rodent, human, definitive host, or intermediate host. Once we know the host, the rest of the clues behave much better. Host first, site second, intermediate host clue third, species last. That order keeps cestodes from turning into a ribbon-shaped identity crisis. When sorting these parasites, start with the host, then use the anatomic site and intermediate host clue to narrow the options. Horse plus ileocecal region and an oribatid mite points towards Anoplocephala. Young grazing ruminant plus broad intestinal proglottids and an oribatid mite points towards Moniezia. Bird or poultry plus an arthropod intermediate host points towards Raillietina. Rodent or human context, especially with a direct life cycle exception, points towards Hymenolepis. These examples all place the animal on the definitive host shelf, and the adult cestode lives in the intestines. However, with Tinea, the sorting question changes. The animal in front of us may instead be an intermediate host carrying a larval cestode in its tissues. That's why the anatomic site is so important. The larvae are not confined to the intestines. First, the equine tapeworm, Anoplocephala perfoliata. Anoplocephala roughly means unarmed head, reflecting the absence of a rostellum with hooks. Perfoliata means appear to be piercing through. The name may or may not have been chosen as a warning about intestinal perforation, but it creates an ominous clinical echo for a parasite capable of severe disease at the ileocecal junction. The scolex has 4 suckers and 4 distinctive lappets, with 1 lappet positioned behind each sucker. These lappets distinguish A. perfoliata from other equine tapeworms, including Anoplocephala magna and Anoplocephaloides mammillana. Our AI-assisted lappets are not exactly drawn to scale. Apparently, even parasite anatomy appreciates a flattering camera angle. Horses become infected by ingesting oribatid mites while grazing. Adults preferentially attach near the ileocecal junction, where heavy infections can cause inflammation, ileal impaction, obstruction, intussusception, and colic. The lappets identify the species. The ileocecal site helps explain the disease. Horse, ileocecal junction, oribatid mite, 4 scolex lappets, and possible colic. Now consider Moniezia species in ruminants. The name itself offers little biologic help, so use the pattern. Young grazing ruminant, small intestine, oribatid mite intermediate host, and large broad proglottids that are wider than they are long. The eggs are angular and contain a characteristic piriform apparatus around the oncosphere. These features identify the general Moniezia pattern, but species-level identification may also use the host, egg shape, proglottid morphology, and arrangement of interproglottid glands. Other ruminant tapeworms can look similar, so one clue should not carry the entire diagnosis. Most Moniezia infections are mild, although heavy burdens in young animals can contribute to intestinal disease or poor health. So remember, young ruminant, small intestine, oribatid mite, broad proglottids, and angular eggs with a piriform apparatus. This slide is here to prevent a very common trap. Anoplocephala and Moniezia both use oribatid mites, which are tiny free-living soil or pasture mites. So yes, the intermediate host clue is shared, but the mite alone is not the diagnosis. If the host is a horse, the pattern supports Anoplocephala perfoliata. If the host is a ruminant, it supports Moniezia. The shared clue is the mite, the deciding clue is the host. In other words, don't let the mite hijack the diagnosis. It is an important supporting actor, but not the star of the episode. Now we reach the important Tinea pivot. With Tinea, the animal in front of us may be a definitive host carrying the adult in the small intestine, or an intermediate host carrying the larval metacestode in tissues. In the definitive host, we think about adults, eggs, or proglottids in feces, and a usually relatively quiet intestinal infection. In the intermediate host, we think about larval stages in the muscle, brain, liver, serosa, and other tissues, along with lesions, necropsy findings, imaging, or meat inspection. This is the role reversal that makes Taenia so high yield. The adult in the definitive host is often a quieter story. The larval metacestode in the intermediate host may be a real troublemaker. Tapeworms can be surprisingly considered guests in the definitive host and much less polite in the host they are trying to leave behind. First, the human livestock Taenia group. Taenia saginata is the beef tapeworm. Humans are the definitive hosts, cattle are the intermediate hosts, and Cysticerci develop in bovine muscle. These larvae are called Cysticerci. Cysticercus comes from the Greek roots meaning bladder and tail, reflecting the bladder-like larval form. The adult scolex is classically unarmed. Taenia solium is the pork tapeworm. Humans are again the definitive hosts, pigs are the usual intermediate hosts, and Cysticerci develop in porcine muscle. Here, the adult scolex is classically armed. The major board and One Health pearl is that humans can also become intermediate hosts after ingesting eggs, leading to cysticercosis. So for T. solium, do not stop at intestinal tioniasis. Ask whether the human is serving the wrong role in the life cycle. Taenia asiatica is a lighter mention but worth knowing. Humans are the definitive hosts, pigs are the intermediate hosts, and the larval stage is associated mainly with pig viscera, especially the liver. The adult is often described as more saginata-like, with the rostellar ones making it a favorite board detail species. High yield contrast: cattle plus muscle points towards T. saginata, pig plus muscle plus human cysticercosis risk points towards T. solium, and pig plus viscera or liver points towards T. asiatica. Now for the canid livestock Taenia group. Taenia multiceps is the dramatic teaching moment. Adult worms live relatively quiet in the canid small intestine, but sheep and goats can develop cianurus cerebralis in the central nervous system. That produces cianurosis, also called gid or sturdy, with circling and other neurologic signs. This is a lovely, if slightly rude, reminder that the larval stage in the intermediate host may be far more damaging than the adult in the definitive host. Taenia hydatidina uses canids as definitive hosts and a range of ungulates as intermediate hosts. The larval stage is called Cysticercus tenuicollis. After hepatic migration, Cysticerci are found on the serosal surfaces such as the omentum or mesentery. Think liver migration, then abdominal serosal cysts. Taenia ovis also uses canids as definitive hosts, but sheep and goats carry Cysticerci in the skeletal and cardiac muscle. Clinical disease is often limited, but the economic consequence matters because carcasses may be trimmed, downgraded, or condemned. And finally, the honorable mention is Taenia crassiceps. Its usual cycle is canid definitive host and rodent intermediate host. But the proliferative Cysticerci make it memorable, and it can occasionally cause aberrant host disease in dogs and people. Now to avian tapeworms, Raillietina species. Adults live in the small intestine of poultry and other birds. Depending on the species, beetles or ants serve as intermediate hosts, and birds become infected while foraging and eating those arthropods. Diagnostic clues include egg capsules or proglottids in feces. Remember, bird host, small intestine, arthropod intermediate host, Raillietina. A quick etymology aside, eponyms can be historically meaningful and biologically unhelpful at the same time. Raillietina honors Alcide Rieu, a French veterinary parasitologist, which is lovely for history and less helpful when identifying a parasite before coffee. His surname appears in several other genus names, including Raillietina for a mite, Raillietia for a pentastome, and several nematode genera. Therefore, pay close attention to the exact spelling and context whenever Raillier's name appears. Hymenolepis nana is the dwarf tapeworm. Nana means dwarf, which is a pleasantly honest species name. Most cestodes require an intermediate host before returning to a definitive host. H. nana breaks that pattern. Its eggs can be infectious when passed, allowing a direct fecal-oral life cycle. An internal autoinfection can prolong or intensify infection within that same host. An arthropod intermediate host may still participate, but it is not required. That is an important exception. By contrast, Hymenolepis diminuta, the rat tapeworm, follows the more typical indirect pattern and requires an arthropod intermediate host, often a grain beetle. Humans may become accidental definitive hosts after ingesting the infected arthropod. So the useful contrast is this. H. nana can complete its cycle directly and can auto-infect. H. diminuta requires the arthropod bridge. Let's bring the patterns together. In horses, think Anoplocephala, perfoliata, ileocecal junction, oribatid mite, four-scolix lappets, and possible colic. In young grazing ruminants, think Moniezia, small intestine, oribatid mite, broad proglottids, and angular eggs with a piriform apparatus. In birds, think Raillietina, small intestine, and an arthropod intermediate host. In the rodent-human context, remember Hymenolepis nana as the unusual cestode that can complete a direct life cycle and produce autoinfection. For Taenia, remember the host's role. The definitive host carries the adult intestinal tapeworm, while livestock and other intermediate hosts carry larval metacestodes in tissues. Those larval stages can cause the greater clinical or economic damage, including neurologic disease, meat inspection losses, and carcass condemnation. The consistent approach is identify the host, determine whether it carries the adult or larval stage, locate that stage, and then use the life cycle bridge to narrow the species. Once those pieces are organized, the names stop behaving like random ribbons and start behaving like biology. Thanks for watching. I'm Christopher Lee, and what you know today protects everyone tomorrow.